Good morning. Today we will be starting the first lecture of metabolic engineering course. Thank you for registration. Thank you for taking this course. So, this course will be taught by me myself and Professor Pinaki Sir from Biotechnology department. We will start with the introductory lecture, basic introduction about metabolic engineering, how the course will go. Overall we will give you an idea what will be covered in this course, and we will take this introductory course into following points like course outline, what we will be learning from this course in several weeks, and then followed by the principles of metabolic engineering, what are the steps followed in metabolic engineering, and then followed by importance of metabolic engineering, why people do metabolic engineering, what is the need for metabolic engineering, that will be discussed in a broad perspective, and then followed by challenges of metabolic engineering. So, what are the problem, what are the issues when you do metabolic engineering, and then finally the application of metabolic engineering we will be learning in this lecture. So, the textbook we will be following, the name of the book is Metabolic Engineering Principles and Methodologies, and this book is actually written by Gregory Stephanopoulos from MIT USA, co-authored by Aristodoy and Jen Nielsen. So, this was published in 1998 and is considered as one of the best book for metabolic engineering. So, in this course myself and Professor Pinaki Sir from Department of Biotechnology, IIT Kharagpur will be teaching this course, and we will cover mostly following topics. It will start with introductory to metabolic engineering, basic concepts, scopes, and then application overview, for example, cellular transport processes, fueling reaction, that will be covered in first week, followed by second week we will have some introduction to MATLAB, and then cellular metabolism overview, the biosynthetic reaction, polymerization, growth energetics, regulation of metabolic pathway. Further, on this third week we will talk about reconstruction of genome scale metabolic network. This will have many subtopics as well, followed by we will have the metabolic flux analysis and metabolomics, where we will learn about flux balance analysis, flux variability analysis, and then flux map, and also we will learn about metabolomics. Then on the fifth week we will learn about experimental determination of fluxes, that is through isotope level substrate, isotope mapping matrix, isotope distribution vector. Those are the subtopic you will be learning. Then on week 6 we will be learning about application of metabolic flux analysis. On seventh week we will learn about experimental tools used for engineering metabolic pathway, and then how to control gene expression, genome editing tool, adaptive laboratory evaluation, and reverse engineering. And on the last 8th week we will learn about some example of manipulation of metabolic engineering, bioenergy, bioremediation, healthcare, and agriculture. So, we will cover as much new topics toward the end where metabolic engineering is applied. So, we will start with some basic introduction, learn about metabolic pathways, and then we will go to advanced topics like genome scale model, metabolic flux analysis, then experimental determination of flux analysis like 13C MFA, then application of metabolic flux analysis. So, and also advanced genome editing tool which is used in synthetic biology. You will also get an idea how you can actually apply this in metabolic engineering. So, come to the basic, like what is metabolic engineering. So, many of you are actually learning for the first time, have no idea what is metabolic engineering. So, I have defined in such a way that metabolic engineering is a process for modification of specific biochemical reaction of the organism, so as to produce required amount of the desired metabolite through recombinant DNA technology. Considering advantage over the other chemical synthesis route, this area of biotechnology is likely to revolutionize in future. So the main idea about the metabolic engineering came that you have to produce some metabolite or a compound, or it can be biofuel, it can be some chemical, industrially important chemical which you want to make in the industry. So it has a huge potential in the sense that most of the chemicals you get from raw petroleum. So most of the chemical which comes from raw petroleum are actually petroleum derived. So to make this environment friendly, because more you depend on the petroleum, so more carbon will be deposited in the atmosphere, and that is a real concern in today world. So if you can produce this chemical or biofuel from microbe by using metabolic engineering, then it will be environmental friendly. And the way it is done is basically because of the revolution in genetic engineering. So because we have a major development in genetic engineering, that is the recombinant DNA technology, you can actually make any chemical from the microbe. Once biochemical reactions are targeted or identified or established, then molecular biology tools are applied in order to amplify, inhibit or delete corresponding enzyme. So first you have to identify the biochemical reaction from which you want to improve the production, and then the metabolic engineering is applied. These involve 4 steps, broadly classified into 4 steps, that is design, build, test, learn. So this is the most latest routine followed in industry and as well as in academic labs. These are the 4 steps one can follow, that is design, build, test, and is also known as DBTL. So DBTL is very popular nowadays for metabolic engineering. So what is DBTL? So DBTL is basically the 4 steps followed in metabolic engineering to design, to build, to engineer the microbes or engineer the biology to produce. As I told, metabolic engineering deal with production of some chemical, enzyme, protein, anything which is industrially important in terms of is a application. It can be a drug also. So it can be chemical, metabolites. You can think of any product or any commercially important chemical which is required in industry and also in daily usage. So to do that, the 4 steps: design, build, test, learn. First, the design is basically you have to select the host. So this is one of the major step in metabolic engineering. You have to choose a host organism. Host organism is the organism that you will be using for metabolite production or chemical production. Any byproduct you want to make, that you have to choose the organism itself. Now these organisms are chosen based on the availability or the knowledge available for that organism. So generally this can be yeast or E. coli, because yeast and E. coli are very well known in terms of their molecular biology tool, in terms of their genetics. We know most of the things, not all of them, but approximately around 50 to 60% of the organism is known, because it has been studied for several decades. For yeast, I am talking about model organism like Saccharomyces cerevisiae, and the yeast is also metabolically engineered to produce different chemical used for bioproduction, and then E. coli is also used. So mostly these host organism can be these 2 organisms, Saccharomyces cerevisiae and E. coli. And then you have to select the pathway. The pathway is selected based on the molecule you are targeting. Suppose you want to make succinic acid. Suppose you want to make some molecule which is produced in the organism but not in a great amount. So the metabolic engineering deal with production of the chemical in a huge amount, not just if naturally it is producing, it may not be sufficient for industrial production. So for that you have to increase the production. Either you have to increase the production, or you have to make a chemical which is not produced by the organism. That also you can do. So both way you have to, if you want to improve the production which naturally the organism is producing, or in other way you can. So this is the yeast. You choose this host organism, and then you choose for the metabolic pathway. Suppose I look for some amino acid, so then to improve the amino acid like the glutamate. So glutamate you want to improve the production. So, you choose that pathway and then you build some cassette. So, these are the genetic parts you want to put in the host organism. So, this is the native organism. So, you have the native organism here, and then this is the you are building the parts. So, in the building parts is also part of synthetic biology. So, in the metabolic engineering also deal with synthetic biology. Many of you heard about synthetic biology. Synthetic biology deal with different biological parts or the genetic parts. For example, promoter, gene, coding sequence, terminator. So, there are many things in the you can include in the part assembly. So, the part assembly is a process where you design the pathway and then you integrate or you put it in the cell. So, before actually putting the pathway, you actually design it in the computer. Design the, or you have heard about synthesizing the DNA. You can synthesize the DNA, or you can synthesize oligo. You do lot of analysis in designing the oligo. That part that goes into assembly plant is also another research which comes in design the assembly plant is also known as biofoundry. So, biofoundry is another branch of study in the metabolic engineering. They design different biological parts and try to assemble the pathway. Suppose you want to make a metabolic pathway inside the cell which has many genes like 10 genes, 20 genes, that you have to assemble in a proper way. Otherwise, what will happen if you put randomly inside the cell, then the cell may not be effective in producing that or producing those protein from the gene. So, you have to design in such a way that you can effectively produce those protein inside the cell. So, because the gene or the coding sequence you are putting inside the cell, it must be produced, the protein should be expressed from those gene, otherwise there is no purpose, right. So, the main purpose is to actually expose these genetic parts. Genetic parts are basically different gene. This can be gene 1, gene 1, gene 2, gene 3, like that. So, G1, G2 are the different genes. So, suppose the native organism, this is native. So, the native organism don't have those genes. So, you want to set up a very new pathway, that is heterologous pathway. Then you have to take out that gene from different sources and you design those gene, you have to assemble those gene and put it into the host organism, so that that pathway is ready. And therefore, for inserting the pathway you have to use genome editing tool. So, right now the technology, you can insert the gene in the form of a plasmid, or also you can integrate in the chromosome. So, in this course we will be learning about CRISPR-Cas9 tool, where you can insert those gene in the chromosome, so that the host organism is permanently modified. You do not have to do many transformation. So, this way if you design or buy those gene and put it into organism through genetic engineering tool like CRISPR-Cas9. So, here the concept of genetic engineering is coming, where the recombinant DNA technology, where you want to put those gene into the chromosome or in the plasmid form. And once you put this assembly part inside the cell, then your pathway is ready, but you do not know whether the pathway is actually doing anything, because in the cell you are not actually able to look, you can visualize that those proteins are formed or not. You are totally unaware of that. So, to check that, the next step is test. So, test is very important, because test you are actually verifying whether that pathway is actually making any sense or not. Many times what happen, when you put a metabolic pathway, then you may not see any improvement of the chemical. So, the best test is through you have to test through high throughput screening or through measuring the metabolite itself. So, in the spectroscopic analysis, you can measure those metabolites. Suppose you are producing some metabolite, you have to measure it. So, the cell is producing some metabolite, you want to measure it using some analytical method. Maybe you can use a GC mass or LC mass, or you can study the expression data, RNA-seq data, or gene expression data, to see whether the inserted gene is expressed inside or not. So this is a checking. If you are checking that whether your heterologous genes are expressed or not, that you have to verify. Otherwise, you will be in a you will not be able to know what is happening inside the cell. So proper checking is needed. So they then learn after you test, after you verify that the metabolite is forming, and then you have to see whether how the carbons are flowing inside the cell. The metabolic flux analysis here is actually dealing with that. So I will be going into the metabolic flux analysis in detail in our lecture, but just to tell you that the metabolic flux analysis deal with the flow of carbon, the flow of metabolite inside the cell, that is the rate of a reaction. For each and every reaction you can estimate based on the engineering you are doing. The engineered cell can be characterized in terms of the flow of metabolite inside the cell, and if you know that, then you can actually again go back and design. So again you go back. So the cycle, you start with a here, and in the next step you add new genes, and then you test it, that how much it is producing, and then you go look for fluxes. So in the metabolic flux analysis, you determine how much carbon is going in each of the metabolic pathway. So this way, this entire process is known as DBT, that is design, build, test and learn. And today the metabolic engineering are done using these 4 steps. Metabolic engineering has evolved for many years, but right now these are the 4 steps followed to actually do metabolic engineering for microbes for bioproduction. Any kind of product you can manufacture from microbes, provided you have the metabolic pathway. And this metabolic pathway we will discuss more in subsequent lectures, that how you can actually select different metabolic pathway. And then also we can discuss about the spectroscopic analysis, that is a part of the metabolomics. So this goes as a part of the metabolomics. And this is a part of synthetic biology. And this is a branch of study is known as fluxomics. So this the metabolic engineer involve lot of branches of study, as you can understand. It's very interdisciplinary field. And first you have to know the cellular physiology, the cell physiology. The cell physiology you have to know. The knowledge of this thing are required for better metabolic engineering. If you know all these 4 parts, then you are a better metabolic engineer, but it is not necessary that you should know each one of them. So you may not be familiar with metabolic flux analysis, but still we will be able to do metabolic engineering. Suppose you don't know this spectroscopic analysis, but still you will be able to do metabolic engineering, provided you outsource the measurement to another vendor or another lab. So this 4 area can be combined together for effective metabolic engineering. So in the next step, I will like to tell that the microbial cell is a chemical factory, because the metabolite inside the cell are actually you can consider as a chemical, but the amount which is present inside the cells are very small. So that is the only difference. If you consider the cell as a chemical factory, and these chemicals are actually produced inside the cell in very small amount, in microliter. So the metabolic engineering, the purpose of metabolic engineering is actually improve the production of this metabolite, that is A, B, C, D, E. These are the metabolite inside the cell. So S is basically a substrate. Substrate, it is going inside, it can be glucose, galactose, any kind of substrate the cell is eating up, and then producing a product P1 and P2. So this product can be ethanol, succinate, any and these are the product you are looking for basically. Here it can be P3. So many product you can make from out of the cell. and suppose you want to increase the production of P, then what happen, you should look for this pathway. So, one particular pathway you have to look at, that is this P2 I want to improve, and P1 P3 you can remove, I do not want, because I only want P2. So, this is the strategy used in metabolic engineering, that when you target a given metabolite, that is P2, and then you look for those metabolic pathway which metabolic genes. So, these are basically metabolic genes which are catalyzing reactions like S to A to B, B to D, and D to E. So, these are actually catalyzed by enzyme 1, enzyme 2, enzyme 3, like that. So, we have a 1, 2, 3 enzymes which are actually involved in forming this final product, and this enzyme needs to be formed inside the cell. The enzymes are the bottleneck. If the enzymes are produced in enough amount inside the cell, then you can actually make more product. So, the product will go up. So, what do? You actually improve the enzyme production inside the cell and try to channelize more carbon along this pathway. So, this is what it is followed in metabolic engineering. You try to get more enzyme inside the cell. How do you get more enzyme inside the cell? What do you have to target? It is very easy, because of the genomics we know the sequence of these enzymes. So, inside the cell you know which gene is actually correspond to this enzyme 1 and enzyme 2 and enzyme 3. So, genomics, because of the sequence of the genome, you know which are the genes are actually correlated with this enzyme 1, 2, 3, and this enzyme you can easily improve the production inside the cell by increasing the expression of the gene. So, you want to try to improve the expression of the gene. How do you improve the expression of the gene? It is also through molecular biology. Here the genetic engineering playing a role. Once you identify the pathway, and then you look for the enzyme which you want to improve inside the cell, and for doing that you need recombinant DNA technology or genetic engineering, where you actually upregulate those gene 1 if this enzyme correspond to gene 1, gene 2 and gene 3, assuming that each enzyme has a 1-to-1 correspondence, that enzyme 1 correspond to gene 1, then gene 2 correspond to enzyme 2, gene 3 correspond to enzyme 3. So, this way if there is a mapping like that 1-to-1 mapping, then by just upregulating this gene, so you try to upregulate this gene 1, gene 2, gene 3, you automatically actually get more enzyme, more proteins are there, so that the reactions can be much more favorable. So, more you have inside the cell, in the sense that more metabolite B will be there, more if you have enzyme 2 more produced in the cell, then you have more metabolite D, and then more metabolite D, and finally this metabolite E is exported outside the cell in the form of a product. So, this is the way you can actually improve the production of certain product you are looking for. So, this gives you an idea what is the basis of metabolic engineering. So, metabolic engineering is also treated as a directed evolution strategy. The directed evolution strategy where first you should know what is evolution. So, evolution is the sequence of events involved in the development of a species or a taxonomic group of organisms. So, it is basically a sequence of event where the species is actually evolving over a time, over several years, so that it can perform a particular function. So, every cell, if you see in the nature, has developed certain function, certain characteristic, and that is not a one day phenomenon. It has actually evolved for several years to reach there. So, this is a very well known evolution, you already know. I do not have to talk much about it, but metabolic engineering does exactly the same, but it is in a more controlled and a faster way. So, we do not have to wait for several years. So, you have to actually can do it in few days, rather than evolution which has come up for several years, but metabolic engineering you can do it in a more controlled way. So, you can develop new living organism by altering the metabolism and the existing one. In that respect, metabolic engineering can be viewed as a method of in vitro evolution. So, you are evolving the cell in vitro, in the sense that you are doing in a directed in a fashion to achieve certain improvement of some bioproduct. So, where you want to improve the production of bioproduct, as in every engineering field, there is a design and analytical component. So, first you design, and then there is analytical component where you analyze how is the design you have made is actually performing. So, directed evolution is all metabolic engineering also a kind of a directed evolution, where you try to evolve the strain so that you do not have to wait for many years. Historically, the synthetic component, that is design build of metabolic engineering, appeared fast. So, in the metabolic engineering, the synthetic component actually came fast, where you can apply molecular biology tool. The main enabling technology is the actually genetic engineering technology, that through which you are actually adding new genes. So, in this diagram you can see that this is suppose you consider this as a E. coli cell. E. coli. Why you have chosen E. coli? Because E. coli is easy to handle. You know most of the things in E. coli. Most of the when I say that most of the thing you know, that means you know most of the genes in E. coli. Not all of them. E. coli is the most well studied organism. One of the most well studied organism where you know most of the molecular biology tool, and you can actually add new genes from different sources, like you can from plant you can get a gene. So, you can insert the gene in the inside the cell from the plant. Also, you can from human being, from other microbe, you can insert, or the other mammalian organism also you can have those genes. So, through genomics, because of the revolution in genomics also, you are able to do this, because a lot of sequences are available right now. If you go to NCBI database, you will get several hundred organisms are available, whose metabolic pathway you want to put it in E. coli, because that particular product you want to make inside the cell. For example, I give one example, like you have heard about anti-malarial drug, right? Artemisinin is actually a anti-malarial drug which is used to treat malaria patient, and also we saw a Nobel Prize for this molecule, the artemisinin drug. The person got the Nobel Prize for extracting this compound from this plant. So, the plant is used to actually extract the drug, but this plant is actually not very easily available. So, that's why. So, one scientist what he has done, he actually inserted the gene which is required for making the artemisinin drug, and put it into a microorganism, and try to make that compound through genetic engineering. So, through genetic engineering he inserted those metabolic pathway for the production of the drug. So, this way you can actually design the cell using the synthetic biology, or the genetic engineering tool can be applied, where you can include 2 or more different sources. You can even include gene from 2 different sources. So, 2 or more different sources, you try to insert the gene in the microorganism like E. coli, and try to generate the compound. For example, the artemisinin drug was produced from yeast, Saccharomyces cerevisiae, not E. coli. So, this is just to tell you that you can get genes from various sources, and during the design you try to insert those metabolic pathway in the chromosome of the cell, so that you have a production. And then the analytical component of metabolic engineering is basically deal with measurement. So, here I am showing one component, that is GC-MS, the gas chromatography setup, for mass spectrometry. So, it has a combination of gas chromatography and mass spectrometer, where you try to measure the compound, how much the cell is producing. This is a very important technique we use in metabolic engineering, where you try to measure the amount of product the cell is producing, and this is a check, primary check, which you can actually know whether your metabolic pathway is working or not. So, learning this is also a part of a metabolomics, where you try to actually measure the metabolite, not all the metabolite inside the cell, but some of the metabolite you can measure using the GC-MS. So, next we will come to another interesting topic, that is the cellular metabolism. The cellular metabolism, you have to understand like how the cell is actually producing different metabolite inside the cell, that you have to understand. Here you can see that different this is the central metabolic pathway which is going straight, that is the carbohydrate metabolism. Like you suppose the cell is going on glucose, then the glucose enter from this pathway, then it go to TCA cycle, and then various amino acids are produced. Then we have lipid metabolism, nucleotide metabolism. So, these are the blocks which are present in the your meta. Is a very well connected network, very complex network. Is not very easy to actually understand how they are actually interconnected. So, for that Professor Pinaki sir will be talking more about different metabolic pathway that is involved in the metabolic reaction, involved in energy metabolism, the amino acid metabolism, nucleotide metabolism. So, this pathway you will be learning in this course to some extent, but the basis of it, this entire metabolic pathway you can actually derive from the genome sequence. So, this is the technology which is available. This is a part of systems biology. So far I told about synthetic biology, where you can integrate gene from any other sources. Now you coming to systems biology, where you can actually identify different metabolic pathway from its genome sequence. So, if you know the sequence of the organism, then you would be able to actually identify what are the metabolic pathway this organism can have. This is the biggest information you have when you start doing metabolic engineering, because if you know what metabolic pathway the cell has, then you can do metabolic engineering much more easier. So, this information is very much available for E. coli and yeast, Saccharomyces cerevisiae, and many other non-conventional yeast also we have this information available. So, what you understand from the cellular metabolism? The cellular metabolism is a very complex network which are interdependent. You can see that each metabolite is connected to each other, very much connected. The metabolome is basically all the small molecule of a cell. So, the cell may have a lot of metabolite that constitute the metabolome of the cell. The metabolic network are defined by pathways. The metabolic network is basically each node is basically a metabolite, and the connection between the two metabolite is basically a reaction. So, this is the understanding. This network is actually any kind of network has two components, one is node and one other is edges. Edges are basically the reaction. So, the metabolic networks you can identify this network directly from the genome sequence, because of the well known technique which are available, which you will learn in this course, that is basically part of systems biology. So, in this course we will learn little bit about systems biology, where you will be able to actually identify this kind of metabolic network or metabolic pathway directly from the sequence. And in the flux you can also measure. Once you have the metabolic network, you can actually calculate the flux. Flux is the rate of turnover of molecule through a pathway. Suppose this reaction, there is a reaction going from here, and now we want to know how much carbon is flowing in this reaction. So, in this reaction, that is also you can calculate and identify how much carbon it is going through. And the flux is regulated by the enzyme in a pathway, and this flux is also regulated, and that's why you need to learn about the regulation of the metabolic pathway. So, in this course we will also learn about little bit about the regulation of the metabolic pathway, how much carbon it is flowing and how much it is regulated. That also you need some idea for doing the metabolic engineering, not just the pathway. You also need to know how this metabolic pathway is regulated inside the cell. This way you would be able to actually have an idea how the carbons are flowing inside the cell, because it is very well connected. So this also you will learn about in detail in subsequent classes. So the metabolic engineering is like managing a traffic. This slide gives you an overview of what is metabolic engineering. So in the left hand side you can see that that we have a metabolite, and then there is a reaction. This metabolite is converted into this compound, that is from glucose to D-fructose. And then each metabolites are connected through a reaction, and there is an enzyme. So this is the enzyme for this reaction, this is the enzyme for this reaction, like that they are connected in a network. It is even compared to traffic. Nowadays if you see the Google map, if you open a Google map, you will see that which roads are actually congested. So the red regions are actually congested, that is there is a traffic jam. So we don't go in that pathway. Because of the Google, you get an online distribution of the traffic jam on the road, and this may happen similar. This thing you can see in the metabolic network where the carbon and atoms are the people. So you can consider the traffic compare it to the metabolic network, and the metabolites are their location, and enzymes are the road and the railway they travel on. So enzymes are basically the road. So if you don't have enzyme, then what happens? This metabolite may not be converted into another metabolite, and the metabolites are the location. So suppose you want to go from here to here, these metabolites are basically the location, and the roads are basically the enzyme. This way you can actually understand that what is metabolic engineering. The carbons and atoms are the people. So the one people is going through this road, and then going from one place to the other, that is from metabolite 1 to metabolite 2, and then it is going through an enzyme. So this is what the analogy which is used in metabolic engineering to understand it better. Now I take a small example of metabolic engineering of succinic acid. So as you know, the succinic acid is industrially relevant compound where is a very useful solvent in industries. The succinic acid it is used in many purposes. It has a high commercial value, and there are many ways you can make succinic acid. So using metabolic engineering, by engineering the cell also you can make succinic acid. Here you have shown the metabolic pathway for succinic acid. So the sugar is entering the cell, it is coming from outside, going inside the cell, and then it is converted into pyruvate, and then from pyruvate to acetyl coenzyme A, and then from acetyl coenzyme A to it goes to TCA cycle, and from TCA cycle you are getting the succinic acid. So this is the product you are looking for. And these are the intermediate metabolite, like it has first converted into pyruvate, and then should be converted into acetyl coenzyme A, and then it should enter the TCA cycle, and then it produce the succinic acid. Now the question is that if I want to improve the production of, so I want to make improvement in succinic acid production. So what I should do to improve the production of succinic acid? So the straightforward way is actually you improve. Suppose you want to improve the production of enzyme 1, as I told in the previous slide, that the enzyme production should improve. So to improve the enzyme production E1, do you think that the succinic acid will improve? Suppose I increase the production of enzyme inside the cell by upregulating that gene which is responsible for enzyme 1. So do we get the succinic acid production increase, because now enzymes are produced more? This way the question is that if I increase the enzyme 1, should I get the improvement of succinic acid production also as well, directly proportional or not? So whatever your answer, the answer is no. Simply the answer is no, because if you improve the production, increase the production of enzyme 1, the succinic acid production may not go high, because because from pyruvate the amount of acetyl coenzyme A produced from pyruvate it is going to acetyl coenzyme A, and this acetyl coenzyme A is going into many other pathway. You can see that acetyl coenzyme A is going into fatty acid pathway, then it is going to monoturbine, then only small portion of the flux goes into TCA cycle. So, the inside the cell, even though you are producing more acetyl coenzyme A, but only the fraction of acetyl coenzyme A which is produced more is going into TCA cycle, and then inside the TCA cycle small fraction of the TCA cycle flux is going into succinic acid. So, this is a systems biology problem, because the metabolite which is produced inside the cell may not be converted totally into succinic acid, that is the carbon. Suppose the carbon from the sugar is entering the cell, then the moment the enzyme 1 is more, if you upregulate the enzyme 1, then more acetyl coenzyme A will be formed, but the more acetyl coenzyme A may not enter into the TCA cycle, it may go into another reaction. That is the idea. That is why metabolic engineering, when you do measurement, this is a system biology problem. You have to understand with a system perspective, where you have to consider the metabolic network, and you have to see how much flux is actually going into different pathway. So, when you make when you are interested to make some product like succinic acid using biological system, then these are the hurdle you will be facing. It is not very straightforward. And since you are improving succinic acid, you are not happy with the wild type strain. So, the wild type may not produce enough succinic acid. So, that is why you are doing the metabolic engineering. Using bioengineering, you want to upregulate this enzyme 1. So, that is why you need a modeling technique, or the how actually the carbons are flowing and going into succinic acid. So, this problem can be addressed using a systems biology perspective, where you want to understand how the carbons are flowing into different network pathways, that you have to be very clear. So, these are the challenges in metabolic engineering that you have to understand the problem in a systems biology perspective. Not only that, if you know the metabolic pathway and then you want to actually make that pathway inside the cell, for that you need genetic parts. So, these are the genetic parts which are available, the coding sequence, terminator, then plasmid, primer. So, all those things you have to you should know, and this is very much easier when you consider E. coli or Saccharomyces cerevisiae. But if you go to non-conventional organism, then the biological parts are less in unknown organism. So, this the parts you have to know when you do the metabolic engineering, and then not only the parts, you should know the molecular biology tool of that organism. So, whether that molecular biology tools are available, because these genetic parts you have to actually insert in the organism. For that you need the molecular biology tool like transformation, cloning, you would be able to do for that organism, and also you should know the genetic circuit. The genetic circuit is basically the regulation, how those metabolic genes which you are inserting inside the cell are how they are regulated, that you should know. These are the challenges in metabolic engineering in terms of biological parts, and also how these metabolic parts are actually regulated inside the cell once you insert inside the cell, and also their molecular biology tool you should know. So, this if you know all those things, then your metabolic engineering is easy. Otherwise, it can be very much time consuming if you do not know many of these things. The first is the metabolic pathway. You should be knowing the metabolic pathway and also the biological part, and also the regulation of the metabolic genes which you are inserting, or how it will affect when you insert a new gene. So, if you know these three things, then it is easier. Otherwise, you have to do lot of study to understand this thing before starting the metabolic engineering. So, now I come to the need for metabolic engineering. The rapid increase of global population and the living standard combined with limited availability of conventional chemical industry to reduce its production cost follows by detrimental effect of environment make metabolic engineering driven biotechnological manufacturing technology the only alternative and the choice of the future. Because right now as I told before also that we mostly depend on petroleum derived product. So, most of the chemical product you use is actually derived from petroleum resource, and metabolic engineering can help in this regard, because when you do metabolic engineering of the microbes is basically renewable way, which is environmental friendly. This is one of the factor for which the government is spending lot of money. A lot of industries are also spending money to actually make this product available through metabolic engineering. And other thing is basically the cost of production. So, the conventional way what industry is producing this chemical may not be cost effective and also not environmental friendly. So, to overcome these challenges you need metabolic engineering driven biotechnological manufacturing technology that can be the choice of the future and is alternative procedure which you can get it higher production through metabolic engineering. To overcome this challenge, metabolic engineering provides the biotech industry with tool of rational strain design and optimization. This brings about significant shift in manufacturing cost and yield of desired product which is suitable for the environment. So, using the metabolic engineering tool you can actually design the strain. Strain is basically nothing but the microbes which you are actually engineering. You have to optimize that metabolic pathway so that the cost of production decreases. So, a lot of engineering, several rounds of engineering you have to do through DBTL cycle, that is design, build, test, learn cycle to reduce the cost of production. So, one of the cost and another is environmental friendly. These are the two main motivation for metabolic engineering, so that you get more yield, more product you can produce in this way through so that you do not have to depend on the conventional chemical industry, where the conventional chemical industries are actually not environmental friendly. So, you have to save your environment when you do metabolic engineering, you are actually making this product in a environmental friendly way, and also you can improve the cost of production. So, put together these are the challenges or these are the benefits you have for metabolic engineering. And the application of metabolic engineering also you can see that you can make lot of things like you can make solvent, food ingredient, fuel, polymers, lubricant, pharmaceutical, pharma company, flavors, fragrance. So, right now using metabolic engineering, the metabolic engineering is applied in this field where you can actually produce lot of things, not just fuel, solvent, several things can be produced using metabolic engineering. And the main metabolic engineering is also used for fuel production for renewable sources, convert biomass into chemical. It can be used for therapeutic compound. There is a lot of funding from US Department of Energy, and the huge investment came from companies like British Petroleum, Chevron, and then Bill Gates. They are investing lot of money for this kind of product, because this is real alternative to the petrochemical industry. From petrochemical industry also you get most of the compound, but it is not environmental friendly. That is the reason that we are getting lot of funding from other organization who are supporting this kind of production. Also there you can think of many other companies like Amris, LS9, Lygos, Genomatica. They are actually dealing with metabolic engineering. So, some of the industries that some of the success the industry came from by applying metabolic engineering. For example, the Genomatica. So, Genomatica has produced butadiene. Butadiene can be used for many purposes where it can be a chemical feedstock that replaces petroleum based product. It is a precursor molecule for many compound which converts sugars into BDO, that is butadiene through microorganism, and right now it is producing 30,000 tons of renewable BDO per year, and it is actually benefits for local agricultural economy. A lot of people are also employed, and this is the Genomatica company is based on California, and the company which is actually producing in Italy, and the plant has been opened in 2016. Many other products, for example, artemisinin, which I have already told, it is a drug we use for malaria treatment, is also produced from Saccharomyces cerevisiae, where it took almost 10 years. Right now, the company which is actually involved in production of artemisinin is the Amaris company, and this product took almost 10 years, which goes from laboratory scale to the industrial scale, and the project cost around $50 million. And then we have the PDO, is actually a fiber used for many purpose, which you can make from it took almost 15 years to actually make this compound available in the market, and the cost of the project is around 130 million, and right now the product is manufactured from DuPont. DuPont is actually making this compound. In conclusion, the metabolic engineering is actually a 4-step process, that is design, build, test and learn. So, metabolic engineering, as you know, is a directed evolution strategy. So, you are evolving the strain in a smart way, in a less time. You are actually through engineering technique, you are actually evolving the strain much quicker way, so that you can produce your desired product. And also you have seen that the cellular metabolism is a complex network, where the metabolites are interconnected, and the reaction enzyme, all these are actually very well connected, and the computational modeling is required, because the production of metabolite is a systems biology problem. How these reactions are interconnected, which I have already told you, need computational modeling. This is an invaluable step in determining the optimum engineering to reach desired product. Suppose you want to improve the desired product, then you have to do some kind of computational modeling to understand how the carbons are moving, or the flux is going from one reaction to the other. And finally, we also learned what are the application of metabolic engineering, and many area metabolic engineering is used, where you can see the improved production of certain chemical or bioproduct, which is helpful for the society. So, these are the references. You can learn about the fundamentals of biochemistry by Boyd Wyatt, and also you can learn about genes by Benjamin Levin. Also for computational analysis of biochemical system, also you can learn this technique from the book given by Everard and Wyatt. Thank you.